Hello, good morning, Year 9. Welcome back to the Plants unit. Today we're looking at the question, how do plants control water evaporating from leaves? As always, please can you put the driving question at the top of your page, make sure you've got the date. This allows us to keep our work nice and organised, so when we're looking back over it, it'll make a little bit more sense. Our keywords for today's lesson are photosynthesis, stomata, water, carbon dioxide, H2O, turgid, flaccid, and aperture. Okay, so these last three words, I suppose you can use to describe the stomata, parts of the stomata, and the guard cells. We'll be using these in context, and that'll make a bit more sense later on. You may have guessed already, this is about the stomata, today's lesson. Right. What are we learning about plants for? Well, almost every ecosystem and animal depends on plants for energy. If we understand how plants work, we can improve the yield of crops and help protect our environments. Your starter is, what are stomata? You don't have to write these down, just maybe think of a quick answer in your head. How could you describe what stomata were? And what is the word and symbol equation for photosynthesis? So again, if you've got a scrap piece of paper available, might be worth jotting these down. Otherwise, don't worry too much, but see if you can think of what the reactants and products for these are. Please pause the video. When we come back, we'll go through the answers. Okay, welcome back. These are stomata. If you were to look closely at the top, especially bottom of the leaf, you will notice that there are these bean-looking things. These are the stomata. These are openings into the leaf, which allows gases in and out of the leaf, especially carbon dioxide in and oxygen out from photosynthesis. It also is how water evaporates from the leaves. And we're going to look at that in a lot more detail today, but basically we're looking at stomata today's lesson. In more detail, stomata. Stomata means lots of them. A stoma could be just one, by the way. So if I ever say, look at that stoma, it just means one stomata. Right, what are stomata? Stomata are holes in the surface of the leaf. Either side of the stomata are two guard cells, which control whether the stomata are open or shut. Okay, we've seen this diagram before in the previous lesson. What I'd like you to focus on right now is the fact that when a stomata is open, the guard cells are bent, and when the stomata is closed, they are kind of pressed together, and there is no opening on the inside. You can't get through. So this is when a stomata is closed. So when the stomata is open, you can see in brackets it says swollen here and shrunken here for when it's closed. We're going to look in more detail in just a bit about what's happening there, but it's all to do with the vacuole and the size and the shape of the cells as well. Okay, but the change of the guard cell's shape changes whether the stomata is open or closed. There we go. Second half of the starters is word and symbol equation for photosynthesis. You've got the reactants for photosynthesis is water and carbon dioxide, the products being glucose and oxygen. The whole point of photosynthesis is glucose, but it also produces oxygen as a byproduct. The reactants are the things that you start with. You need water, but especially you also need carbon dioxide. Now this is a gas that the plants get from the air through the stomata. The symbol equation is 6 H2Os plus 6 CO2s makes 1 C6H12O6 plus 6 O2s. Okay, hopefully now we're quite familiar with photosynthesis. Again, it's an endothermic reaction. It requires energy. That's why I've written like there. Okay, so stomata open to allow gas exchange, to allow carbon dioxide into the leaf and oxygen out. We've seen that carbon dioxide is a reactant. Photosynthesis needs CO2 and it produces oxygen, which is a gas. Some of you will know that our atmosphere contains roughly 20% oxygen, which is how we can breathe and how we can get the oxygen for respiration, which is what we need to do. And we get it from plants. Plants don't realize that they're doing this, of course, but they're still doing it, and that's one way that you can remember that oxygen is released through the stomata. Stomata close to prevent too much water from escaping the leaves. Okay, so another thing is that plants need water. Obviously, we've seen some of you who might have looked after plants before, or maybe not done such a good job, you've noticed that the plants go wilt when there's not enough water. We know plants need water, and if the stomata are open, perhaps too open, lots of water can evaporate out of those, and perhaps the plant doesn't have enough. So the plant will prevent too much water from escaping by closing these stomata. There we are, there's a wilted plant for you. And this fact, the fact that the stomata will open and close, depending on photosynthesis, but also depending on water levels in the plant, is what we're going to be looking at today, especially. So, role of the stomata, what are they? And you're going to need to know this for the describe section of today's objectives. The stomata control gas exchange in the leaf. Each stomata can be opened or closed depending on how turgid its guard cells are. In the light, the guard cells absorb water by osmosis. They become turgid and the stomata opens. Okay, so first of all, let's dissect this. Turgid is one of the key words for today's lesson. And what that means is it's, I suppose, full of water, which would make it more solid. Okay, so when the stomata are opened, it is because they are turgid. The cells fill with water and get this rigid shape, and that shape means that there is a hole in the middle that the gases and water can move through. I've got a diagram to help explain that in a second. So in the light, the guard cells absorb water by osmosis to become turgid, and the stomata open. In the dark, the guard cells lose water and become flaccid, and the stomata closes. Okay, so flaccid is kind of the opposite of turgid and means droopy or floppy. And when they droop, that's when they no longer hold this shape, and therefore the gap between the guard cells goes, and then water cannot move. So the take-home message from this slide is that in the light, the guard cells are turgid and the stomata is open. In the dark, guard cells close, and that's because the guard cells are flaccid. Okay. The only way water can be lost from the leaves is through the stomata. Okay, so we know that from the first lesson that the epidermis, which is kind of the skin layer of cells on the leaves, on top of that is a waxy cuticle. Now, some of you will know that wax is waterproof. The fact that it's a waxy cuticle means that the leaves are waterproof. The only place that water will leave the leaf from will be through the stomata. Okay, because it can't just seep through the skin because it's waterproof. The waxy cuticle on the outside of the outer epidermis cells is waterproof. Okay, what's the difference between open and closed stomata? Well, an open stomata has turgid guard cells, and the closed stomata has flaccid guard cells. Okay, this is what that looks like. So again, in the light, the cells fill with water and become turgid. This word, turgid. In the dark, the guard cells become flaccid and the stomata closes. There is an opening between the stomata here, and there is no opening when the guard cells are flaccid. Okay. Notice that I changed the words from last time to turgid and flaccid. We're going to have to get used to using the term turgid and flaccid to describe guard cells. Right, turgid and flaccid, a bit more detail. How can we remember what these words mean? The membranes of turgid plant cells are pressed right against the cell wall. This means they are enlarged and swollen with water. The way to think about this is like a fully inflated balloon, or maybe a football or a bike tire that you've pumped full of air or full of water, and so that it's actually kind of tough. It's not floppy, it keeps its shape. Okay, so just like a balloon that is fully inflated, the stomata form their shape. Okay, so when the stomata are open, the guard cells have this specific shape because they are full of water. This is them being turgid. The opposite is flaccid. So flaccid cells are soft and floppy. They no longer hold their curved shape, and the gap between the guard cells closes. Okay, does that make sense? So because the cells are not full, they press together again. Right, can you please match the stomata shape to the description and the function? Okay, so you've got stomata open and closed, guard cells turgid, guard celled flaccid, and what then that happens. Please read all these boxes, and can you match them all together? Pause the video, we'll go through some answers when you come back. Okay, welcome back. Let's have a look at some answers. Not very much has changed. The second two are the other way around. So these are the answers. The stomata that are open have guard cells that are turgid. This allows gas exchange and water to escape. When the stomata are closed, this means the guard cells are flaccid, so no water or gases may leave or enter. Okay, well done for those people who got that right. Right, again, quick quiz. Which of these is not true for stomata? A, there tends to be more stomata on the bottom of the leaves than the top. B, guard cells can change shape. When they're flaccid, the stomata are closed. C, the stomata allow carbon dioxide to enter the leaf and oxygen and water to leave the leaf. And D, stomata only open at night, so too much water doesn't escape in the day. Which of these is not true? Pause the video, have a think. Right, answers. E, stomata only open at night, so too much water doesn't escape in the day. That's not necessarily true. Okay, stomata will be open in the daytime as well. They have to be open in the day, otherwise there's not going to be any carbon dioxide getting in for photosynthesis. That also means that all of these others are true. Okay, there we are. Right, as we're scientists, we like to test things. How could we test that water has left the leaf? Okay, so I've been telling you that water leaves the leaves through the stomata, but how would we know this happens? Well, we can measure the mass of the leaf. So we would use one of these, a top pan balance, like a kitchen scales basically. And if the mass decreases over time, then we can say that maybe something has left the leaf. In this case, it'd be water has evaporated. If the mass doesn't change, then the water must have remained inside the leaf. Simply, has the mass changed? I'll stick a leaf on the top pan balance, see if it changed. And then over time, would that change? So I've got a question for you. Which would lose the most water? Is it a leaf or an identical leaf covered in Vaseline? Weird thought. But if you covered a leaf in Vaseline, would that lose more or less or the same amount of water than a leaf that's not covered in Vaseline? Well, how could we figure this one out? Well, we'll do an investigation. This is an investigation that we could do in the lab. Unfortunately, we're not doing it at the moment because we're not in the lab. But when we come back to school, we might have the opportunity to do this, and it will be easier to remember, obviously, after we've had a go. So first, measure the mass of both leaves. They may be different, but it's the change in mass that we're looking at, so it's fine. Then hang them out to dry for an hour. Literally stick them on a washing line. Looks a bit silly, but it's how to do it. Then you measure their masses again and compare them. Which one has changed the most? The leaves without Vaseline will lose the most water. Why is this true? Well, the leaves covered in Vaseline will not change mass. Why won't they change mass? Because no water can escape. Even if the stomata are open, they've been plugged up by the Vaseline, and therefore the water inside them will remain inside them. That's the take-home message. If you plug up a stomata, it will not allow water to escape. Another question for you. Are there more stomata on the tops or the bottoms of the leaf? Well, some of those, the keen-eared viewers will have noticed that we have mentioned this before, and I've said that there are more stomata on the bottoms of the leaf. But actually, as scientists, how could we find this out? And how did the scientists find this out in the first place? Well, my question to you is, what could we use to find out? And how could we use clear nail varnish to help find out? So some of you might have done this, used nail varnish on leaves before in the lab, and if you haven't, I'll make sure that we have the opportunity to do this once we get back, because it is quite fun practical. Well, we'll use one of these. What is this? It's a microscope. We've talked about this before. It allows you to see greater magnification objects. So we put the leaf under that, basically, or do it. So we've got ourselves a leaf, and what we do is we cover it in nail varnish. What? Just a fine layer of clear nail varnish. Then, using some sellotape, we peel off the nail varnish and put it on a microscope slide. Okay, so now what we've got is an imprint. The nail varnish, we know nail varnish dries, and it fills in the shape of the stomata, and then when you peel it off, imprinted on the dried nail varnish is an imprint of the stomata. So you've got basically an exact copy of what the leaf looks like. Stick that on a microscope slide, perhaps put a cover slip over the top of that, and then you can view that under the microscope. Okay, and what you could see is something like this. So that's just you moving the slide around, but you should be able to see that you've got the stomata filling up the whole field of view. And actually, clearly, you can count them, literally count them. So 1, 2, 3, 4, 5. 5 stomata in this field of view there. So, again, in words, how could we compare the top and the bottom of the leaf? I've just shown you how to look at one. Comparison, simply just look at the different numbers. You might want to take a mean. Step by step, how would we do it? Well, first, paint the desired side of the leaf with a thin layer of clear nail varnish. 2, once dry, use clear tape to peel off the clear nail varnish and place on a microscope slide. 3, look at under the microscope. 4, change the magnification until the imprint of the leaf surface takes up the entire field of view. The entire field of view. So the whole thing you're looking at is, takes up the whole field. 5, count the number of stomata. So that's as far as we've got so far. Now the next bit is allowing you to compare these things. So using the same magnification, and it's important you use the same magnification each time, repeat multiple times from the same side of the leaf. You could use perhaps multiple leaves as long as it's the same side each time, and then record an average of those. So now you've got an average number of leaves for that side of the leaf. That could be, on average, 5 for the top side of the leaf. Now it's the comparison bit. Ensuring the magnification does not change, this is because we're doing a comparison. Repeat the observations, but for the bottom side of the leaf. Maybe do it multiple times, record an average, and then you can compare the average number of stomata at the given magnification. Okay, so again, this will be a lot easier to remember once we've done this in the lab ourselves, but we are looking at the number of stomata, doing that multiple times, comparing and getting an average, and then comparing that to what you'd see on the bottom. I'd like you to pause the video now, look at A, B, C, and D, and see which order these should go in. You don't have to write anything down other than perhaps the letters on a scrap paper, just to help you remember. Okay, please put these in order, pause the video, and when we come back, we'll go through the answers. Okay, welcome back. Look at this. P first, paint the desired side of the leaf and use clear tape to peel off and put it on a microscope slide. Then B, look at under a microscope, count the number of stomata. A, record an average for doing it multiple times and ensure that it doesn't change, and repeat the observations, and then C, compare the average numbers at the given magnification. So there we are, D, B, A, and C. Right, writing phase. Make sure that you are writing your answers with full sentences, please, and in paragraph, in full paragraphs, please. So, describe section is describe the location and function of stomata and guard cells. Explain is explain how plants control the rate of water loss from their leaves. So, explain what's going on in terms of the guard cells. Use to explain the change in mass of a leaf hung out to dry compared to the same leaf but covered in Vaseline. And 4, write a method to compare the distribution of stomata on the top and the bottom of the leaf. Distribution just means how many of there are in a given area. Okay, please do use the keywords for today's lesson and give yourself about 10 to 15 minutes on this. Once we come, pause the video. Once you come back, we'll go through some answers. Good luck, Year 9. Okay, welcome back, Year 9. Let's go through some answers again. Grab yourself a different coloured pen. Give yourself a tick if you got it right. If you're missing any information from the answers, then make sure that you do add that in. Okay, answers for describe section. Stomata are tiny openings on the tops and bottoms of the leaves. They are significantly more stomata on the bottom compared to the top. When the stomata are open, they let oxygen and carbon dioxide in and water out of the leaves. They close to prevent too much water loss. Guard cells are specialized cells on either side of the opening that change shape to open and close the stomata. To explain what's going on. When stomata are open, water can evaporate from inside the leaves and exit as water vapor out of the stomata. If there is too much water loss, the stomata can close. Perhaps more water is evaporating than can be absorbed from the soil, in midday or in the summer or something. When guard cells on either side are turgid, they are kidney bean shaped, so present an opening between them. To close, the stomata cells become flaccid. They change shape and the holes become closed. Okay, so it's this bottom paragraph really that is answering that. Turgid compared to flaccid, open compared to closed. The U section. A leaf hung out to dry on a washing line will lose mass as the water inside evaporates. A leaf hung out to dry but covered in Vaseline will not lose mass. This is because the Vaseline is waterproof and will plug up the stomata, not letting the water vapor escape. So that's the explanation there. And finally, the combined section. To compare the number of stomata on the top against the bottom of the leaf, we'll use a microscope. First, clean the leaf to remove any dirt that may obstruct the view to the stomata. Then paint the desired side of the leaf with a thin layer of clear nail varnish. Once dry, peel off the nail varnish and place on a microscope slide. On top, place a cover slip. Change the magnification until the imprint of the surface takes up the entire field of view. Count the number of stomata. Using the same magnification, repeat multiple times from the same side of the leaf and record an average. Ensuring the magnification doesn't change, repeat the observations, but this time for the bottom of the leaf, and then compare the average number of stomata at that given magnification. That should tell you that there are more stomata on the bottom, on average. Okay, well done, Year 9. Please can you write your today I've achieved sentence, make sure that you are using the words describe, explain, use or combine to fill in the gap there, and say why. Because I can. Describe the locations and functions of stomata and guard cells. Explain how plants control the rate of water loss from their leaves. Explain the change in mass of a leaf hung out to dry compared to the same leaf covered in Vaseline. Or, because I can, write a method to compare the distribution of stomata on the top and the bottom of the leaf. Pause the video, write that down. When we come back, we'll move on to the next slide. Okay, thank you so much for listening. Well, Year 9. Again, as always, you've got a 10 question show my homework quiz. Please can you make sure that you've completed that following this lesson. Yeah, I'll see you next time. Well done.